Hello everyone, let's get started. Today we're going to be talking about the cytoskeleton. As we learn all about the biology of cells, we sometimes forget about the cytoskeleton because we can't see the cytoskeleton under the microscope with most of the stains that we use every day. The cytoskeleton is really a living, changing scaffolding within a living cell. It helps maintain the shape of a cell, the way that a bony skeleton does in animals. It helps to move things around inside the cell, and it helps to move the cell itself. It's important to remember that only eukaryotes have a true cytoskeleton. Bacteria and archaea have something similar to a cytoskeleton, but it's more primitive and it's composed of different proteins. A true eukaryotic cytoskeleton is composed of three structures: microtubules, microfilaments, and intermediate filaments. Microtubules, as the name suggests, are actually tubes, hollow tubes, and they are made out of a repeating subunit called a heterodimer. Now remember, a dimer is simply a molecule that's made out of two subunit molecules that have come together to work as one. A heterodimer is simply a dimer that is made out of two different subunits that have come together. So the heterodimer in a microtubule is made out of alpha and beta tubulin, two forms of this protein called tubulin. They come together and they work as a heterodimer. They work as the subunit or the monomer unit to build one of these microtubules. The alpha and beta heterodimers actually polymerize into these hollow tubes, and they're relatively large on a cellular scale. They're about 25 nanometers in diameter. Microtubules polymerize out of a very particular structure in a very particular location in a cell. The location is called a microtubule organizing center, or an MOC, and there are typically two MOCs in every cell. We also call these centrosomes, these locations in the cell. Now, inside the microtubule organizing center, inside the centrosome, are physical structures called centrioles. And these centrioles are where the microtubule grows out of as it polymerizes. One of the most important things to understand about microtubules is that they are very dynamic. They are continually growing and shrinking back. In other words, it's not that the cell makes the microtubule and then leaves it. The cell is constantly adding onto it and taking away from it. So it's getting shorter and longer and shorter and longer, just constantly. It's also being redirected from the centriole into new locations of the cell as it is needed to do work. So this means that microtubules have directionality. Remember, when we say that a molecule has directionality, what we're saying is that each end of the molecule looks different. So in the case of a microtubule, we describe a minus end and a plus end. And what that means is that the microtubule can be added to, it can be elongated at the plus end, and it can be taken away from, it can be shortened at the minus end. On this slide, we're looking at a diagram that shows us how microtubules are first created. And as you can see, it all begins with the tubulin heterodimer. So in the drawing, the artist has shown two circles that are attached to each other. One of them represents alpha tubulin and one beta tubulin. So the first thing that happens is these tubulin heterodimers start to be assembled together into short structures that you would call an oligomer. That term oligomer is a generic term. It just means a short, a short structure with few subunits. These oligomers are then going to join together and create what's called a protofilament. That's a longer structure. Several protofilaments will then be joined together into a sheet, and then the sheet will be rounded out so that the two ends come together and join, and they create this tube. So this is how a microtubule is first assembled at the centriole, starting with the tubulin heterodimers, creating the strands, putting the strands into a sheet, and then bringing the ends of the sheet around and connecting them so that you get the tube. Now, once the tube is created, remember, it doesn't just sit there in the cell. It's very dynamically changing. More tubulin can be added to the plus end, or tubulin can be removed from the minus end, so that the microtubule is constantly changing in length. So microtubules are first created in those very special locations called microtubule organizing centers or centrosomes. It's important to remember that these are locations, not physical structures. The physical structure is the centriole. Inside each of the microtubule organizing centers, there is a pair of these centrioles. There are typically two microtubule organizing centers in the cell, and they tend to be located near the nucleus. But at different times during the life of the cell, the microtubule organizing centers themselves can move. This is especially evident when cells divide. If you recall, when you learned about mitosis, you know that the mitotic spindle forms. And the mitotic spindle has to stretch all the way across the length of the cell in order to do its job, helping to separate out replicated chromosomes. So during cell division, the two microtubule organizing centers, the two centrosomes, actually travel to each end of the cell, to each of the poles in the cell. And they take these paired centrioles with them. And then, as microtubules grow out of those pairs of centrioles, they can stretch all the way across the cell. Now, centrioles themselves have some very interesting anatomy. When you look at them, they are composed of 9 sets of triplet microtubules. I'll show you a picture of this in just a minute. Each of these sets of microtubules is at a right angle to the next set, so that you end up with this circular structure made out of short microtubules. It makes sense that the structure that will help produce microtubules actually has microtubules within it, because again, the microtubule is formed by just adding more on, polymerizing more of this heterodimer onto the structure. So we can think about a centriole as what we would call a nucleation site, a place where you can add more tubulin on and create a much longer microtubule. Having the centrioles in the cell, in other words, having these already started microtubules in the cell helps make microtubules much more quickly than if you had to bring them about from scratch. On this slide, in the upper left-hand corner, you can see an image depicting a cell, and a wedge has been removed from that cell so that we can look inside. The purple area represents the nucleus. There's a centrosome right next to the nucleus, and it contains a pair of centrioles. The artist has then taken a centriole out and blown it up for us so we can see what it looks like. This structure, this centriole, is actually made out of short lengths of microtubule. You can see that there are sets of 3. There's 1, 2, 3 microtubules here joined together. Then if I go to the next set, it's shifted to about 90 degrees. 1, 2, 3 here. Then we shift another 90 degrees, 1, 2, 3, so that we create this short, stubby little tube. Now, the black lines that are drawn in that are holding the sets of 3 together, those are just additional proteins. Now, if you look at centrioles under an electron microscope, because you would need an electron microscope to see them, when you look at the centriole head on, in other words, when you look at it on its face here, it looks just like a symmetrical tube. When you look at it along its length, it looks very different. It looks like this. And this EM image is showing us one very important fact about centrioles. Remember, they occur in pairs. There's a pair of centrioles at each centrosome, and the pair is oriented at a 90-degree angle to each other. So one centriole will be oriented in one direction, and the other centriole will be perpendicular to it. Now, when you watch these centriole pairs across time, you can see the microtubules growing out of them and growing out in all directions. That's why having the pair of them oriented perpendicular to each other is important. It allows the microtubules to grow out in all directions in 360 degrees. Here's another image depicting what the centriole pair looks like, and then another EM image showing them as they actually lie in the cell. Again, you can see the triplets of microtubules, and each triplet is connected to the next triplet all the way around the centriole. Let's take a look now at a short little video of microtubules being assembled and dynamically changing within a living cell. This is a time lapse, obviously, but what you're looking at is a pair of centrioles in the center here and microtubules growing out of them. You'll notice that if you pay attention to one microtubule, as I play the video clip again, if you watch one microtubule, you can see it getting longer and shorter and longer and shorter across time. The cell is constantly altering the length of its microtubules because it needs them to do different jobs in the cell at different times. Remember the roles that the cytoskeleton plays in a cell. It maintains the shape of the cell. It helps move things around in a cell, and it helps move the cell itself. In order to do these things, it has to be dynamic, it has to be changing. Microtubules are participating in all three of these functions. They help cells keep their shape, particularly against the kinds of forces that would crush a cell, pushing forces. They also play a big role in shifting things around in a cell. As I said earlier, remember, microtubules form the mitotic spindle that helps move chromosomes around during mitosis. Microtubules are also involved in moving organelles from place to place. Not all of the organelles move in a cell, but some of them do, and they have to be transported. Microtubules are also those roadways or those tracks that vesicles are transported across in a cell. When it comes to moving the cell itself, remember we're talking about structures like flagella and cilia on the outside of a cell, and it is microtubules that make up these structures. So microtubules play a role in all three of the functions of the cytoskeleton. So when we talk about this dynamic nature of microtubules, we give it a name. We call it dynamic instability. Microtubules are not just created and then left there in a cell. They are constantly either growing or shrinking back, depending on the needs of the cell. And the cell has the ability to make this growth or this loss in length happen very, very quickly. Microtubules, in other words, are always ready to shift either towards growth or towards shrinkage in a moment of time. And the way the cell does this is by keeping a microtubule in constant motion. We refer to this as treadmilling. Now we're going to look at some diagrams of treadmilling. It's important to know that treadmilling is an energy consuming process. And instead of using the ATP molecule as the source of this energy, the GTP molecule is used instead. Magnesium also plays a role in this treadmilling process. It's required for treadmilling to occur. Now, each of the heterodimers inside a microtubule is going to bind with two GTP molecules. So the alpha tubulin will bind a GTP and the beta tubulin will bind GTP. And through the hydrolysis of GTP, remember that means breaking the outer phosphate group off of the molecule, a little bundle of energy will be released, and that will fuel the treadmilling process. On this slide, we're looking at a diagram showing treadmilling across time. So if you start at the top of the diagram, you're looking at a representation of a microtubule. Remember, it's made out of individual subunits, and each subunit is composed of one alpha and one beta tubulin bound together. So in this drawing, the minus end is over here, the plus end is over here. Remember, the plus end is the end where more heterodimers can be added on. The minus end is where heterodimers can be lost. So when a microtubule treadmills, we are adding more on and taking off in balance. So the microtubule doesn't change length. It's similar to a person walking on a treadmill but not going anywhere. This microtubule is not going to change in length despite the fact that it's being added to and removed from The reason it doesn't change in length is because those two things are happening in balance with each other. Now, at any moment, at any one moment, a cell can either speed up the addition or speed up the removal, and the treadmill will change. Instead of staying the same length, it will either get longer if this goes faster, or it will get shorter if this goes faster. But under normal circumstances, the treadmilling keeps microtubules the same length. Now let's go and follow what the artist is showing us in this diagram. The tubulin heterodimers that are being added on to the plus end have been colorized. They're green. And the reason that's been done is so we can follow these exact molecules across time. From the start of the diagram to the end, the microtubule is staying the same length. But because we're both adding on and taking off, as you follow the green over time, it's moving. These actual heterodimers are traveling down the length of the microtubule across time until eventually they'll come off the other end. This is treadmilling. The microtubule stays the same length while new heterodimers are added on and old heterodimers are removed at the same rate. This is an energy requiring process. What's interesting is that during treadmilling, the addition of GTP-bound tubulin actually promotes the addition of more GTP-bound tubulin. In other words, when GTP tubulin heterodimers come on to the microtubule, and remember there's one GTP bound to the alpha part and one bound to the beta part, so each heterodimer is carrying 2 GTP with it. As they come on to the plus end, they actually attract more. That's how more and more and more are added to the plus end. And this creates an area on the plus end of the microtubule that we call the GTP cap. Now, in order for a microtubule to grow, to get longer, there has to be plenty of tubulin around. So the concentration of tubulin has to be relatively high in the cell. As long as there's plenty of tubulin around, then there will be plenty of GTP-bound tubulin around, and you'll develop this GTP cap at the plus end. The reason that the plus end is the side where more and more heterodimers are added and not lost is because of this GTP cap. In other words, the GTP cap helps prevent the removal of heterodimers from the plus end. The removal process will only happen at the minus end because there won't be a GTP cap on the minus end. So any growing microtubule is going to have a nice heavy GTP cap at its plus end. The more GTP heterodimers come on at the plus end, the more are attracted, and the cap will grow. Now, remember, treadmilling is going on. So these incoming GTP-bound heterodimers will come on to the plus end, but will then start to treadmill down the length of the microtubule. So GTP will be hydrolyzed as this is happening. There's going to be a boundary, a line almost, along the length of the microtubule where GTP is bound, then hydrolysis is occurring, and now it's GDP bound tubulin. That means if you could stop the treadmilling for just a moment in time, you could see a lot of GTP bound tubulin near the plus end and a lot of GDP bound tubulin near the minus end. Any microtubule that is growing is gaining GTP tubulin faster than it is losing GDP tubulin. There's more GTP tubulin coming in at the plus end than there is GDP tubulin being lost at the minus end. That will allow the tubule to grow. That's what we're looking at here on this slide in this diagram. Now this diagram is intended to be read from the bottom upwards. So what we're looking at down here is a length of microtubule, and they're showing us that this is the plus end, this is the minus end. They've colorized the heterodimers again for us, but this time the green color represents any heterodimer that is bound to GTP. So remember, those are going to be added at the plus end. Treadmilling is going to occur, and they're going to start rolling down the length of the microtubule until they're removed off the minus end. Remember, there has to be a lot of tubulin in the cell. There has to be a relatively high concentration of tubulin available in the cell in order to make these GTP-bound heterodimers. But as long as there's plenty of tubulin, more will be added on this end than will be taken off on this end. So in they come, they get added into the plus end as these green heterodimers. They start to treadmill. GTP gets hydrolyzed. GTP gets hydrolyzed. This is an energy-consuming process. And now the heterodimers change color. They change to the yellow color because now all of these heterodimers, they're no longer attached to GTP. They're attached to GDP now as they continue treadmilling. So there's this barrier, this line in a microtubule between the GTP bound and the GDP bound. As long as, again, there's more being added than taken away, growth is going to occur. And we're going to see this cap get bigger, bigger and bigger. Remember, the presence of the cap attracts more GTP tubulin to the cap. And the presence of the cap prevents anything from being removed from this end. So what is it that allows a microtubule to lose length? Well, the first thing is a lack of tubulin. So the tubulin concentration gets reduced, it gets lowered. That's going to create fewer heterodimers that are bound to GTP. And that, by necessity, is going to deplete that cap. Now, the microtubule will lose length when more GDP-bound tubulin is removed than is being added. So there's more being removed at the minus end than is being added at the plus end. Now imagine this going on. For a while, the cap can just get smaller and smaller and smaller. As the cap gets smaller, though, the plus end is no longer protected from losses. If we lose the GTP cap in its entirety, we've lost the protection from losses at the plus end. So for a while, the cap is there, it's just getting smaller, and the microtubule is just shrinking in its length. There's less cap, there's more GDP being removed from the minus end, and the microtubule just gets shorter. There will come a point, though, when the GTP cap is lost. In other words, as a microtubule shrinks, as the rate of removal at the minus end is greater than the rate of addition at the plus end, the cap will get smaller and smaller and smaller. And eventually the cap will be lost. Once the cap is gone, there's no more protection from loss at this end, and an event will occur called a catastrophe. I love that term. A catastrophe, a catastrophic breakdown of the microtubule. This is what it looks like. Once the cap is gone, now you're not only going to lose heterodimers at this end, but you'll start to lose them at this end as well. And basically the whole microtubule will just collapse. So during catastrophe, not only is the microtubule shrinking, but it is literally just destructing. It's being lost in its entirety. Living cells can control the length of their microtubules in real time. As we saw in that short video clip, microtubules are dynamic. They are constantly changing in length and in direction that they're heading out into the cell. And the cell has complete control over this. And it can control this by controlling how much tubulin is around. It all begins by the expression of these tubulin genes so that the supply of alpha and beta tubulin is constantly changing, and that's going to change whether or not the tubules are simply treadmilling, whether they are growing, whether they are shrinking, and whether they are collapsing.